Curative Therapies for Sickle Cell Disease. We have two dynamic speakers today. Our first speaker will be Dr. Krishnamurti, Professor of Pediatrics at Emory University. He's a pediatric hematologist oncologist with 25 years standing with primary research focus in sickle cell disease. His main interest is clinical and patient-centered outcomes research. He is research funded by NHLBI, NICHD, CDC, HRSA, PCORI, and many others. He is currently PI of the NIH U01 grant for multi-center study of hematopoietic cell transplant and the Sickle Transplant Evaluation Long-Term Late Effects Registry, or STELLAR. Our second speaker is Julie Kanter. She's director of the Adult Sickle Cell Program at the University of Alabama, Birmingham. She works closely with national partners, including ASH, SCDAA, CDC, and NIH. She has specific interests in gene therapy, adhesion therapy, and novel combination therapy. Dr. Kanter received NHLBI funding for dissemination and implementation research to improve access to care for affected individuals with sickle cell disease. Dr. Krish will speak with us today about bone marrow transplant, and Dr. Kanter will bring us up to date on gene therapy for sickle cell disease. So Dr. Krish will be our first speaker. Thank you.
Good afternoon. Thank you, Dr. Pace and Jessica Homa for having me on. It's a pleasure, and so I'm going to share my slides here, and I hope everybody can see and hear me. So it's my pleasure to talk about curative therapy, focusing on really bone marrow transplant, hematopoietic stem cell transplant, how we have come from promise to reality. Over the course of the next 20 or 25 minutes, I hope to talk to you a little bit about why we would consider curative therapies and when. What are some of the different conditioning regimen by which we prepare the patient's body to receive the bone marrow? And what are the different types of donor options we have? What are outcomes with these donors? And then pivot a little bit to the ethics of bone marrow transplant, something about cost effectiveness, and then really take a look at what this actually means for the rest of the world beyond the vantage of the United States, and then leave you with some take-home messages. In my presentation today, I will start. So why consider transplantation for sickle cell disease? The answer is actually fairly straightforward. It's a horrible disease with substantial morbidity and premature mortality. This paper from Dr. Michael DeBon in Blood from a couple of years ago shows that there's a 20-year loss of life expectancy in adults with sickle cell disease. So that's the main thing, is that we're offering curative therapies to patients with sickle cell disease because it is a life-threatening illness and a quality of life destroying illness. This audience
really lied to Congress. Usually what they say is, can you report at 8 a.m.? That would be the...
Okay, so I'm not totally sure what's going on there. I'm just going to put this slide on. Okay, so transplant for sickle cell disease offers a very complex risk-benefit paradigm. On the one hand, you can cure a deadly disease. On the one hand, there is a possibility of living free of the risk of complications of sickle cell disease. On the one hand, there is improved survival long-term. On the other hand, there is a risk of death as a result of the transplant procedure itself and its complications. On the other hand, there is risk for treatment complications that can cause substantial morbidity and then long-term adverse events. So this risk-benefit paradigm is further modulated by the age of the patient to see what kind of donor options are available. And as Dr. Kanter and others have been working on, what are the other treatment options that may be able to improve outcomes without doing a transplant? So in general, the the commonest reasons why somebody might consider transplant is that they have vaso-occlusive pain that has become intolerable, that they've had stroke and have had recurrent transfusions, and they have complications from blood transfusion, and the possibility that stroke might occur, or they have recurrent acute chest syndrome with serious pulmonary complications.
Really lied to Congress.
So what are the outcomes of transplantation for sickle cell disease? It turns out that younger age at transplantation predicts good outcomes when you do a transplant from a sibling who is identical on the human leukocyte antigen panel, HLA. And look at this here. In the blue here is a child under the age of 5 years. In green here is child 6 to 15 years, and in red is more than 15 years. So overall survival under the age of 5 is 99 plus minus 1%. You don't have to be a statistician to know that we're at the margin of being able to make this any better. But when you go down to 10 years later, there's almost a 10 to 12 point drop in overall survival. Similarly, the event-free survival is 96 plus minus 2% if you're under the age of 5, and it's really 12 to 14% less if you wait another 10 years. So this is the picture that from Dr. Kebele's paper, which shows the same thing, is that the event-free survival, overall survival, event-free survival, all are better when you have a matched sibling donor and you do the transplant early, and as do all of the complications of transplantation are less when you do the transplant early. So here is a risk prediction strategy, which basically takes two things into account: age and type of donor. So the younger the patient, if the patient is less than 12 years old, there's an age score of 0. If you have an HLA matched sibling donor, you get a donor score of 0 for a total score of 0 and excellent outcomes. Again, if you're younger, but you have a matched unrelated donor, then you're an intermediate risk, and then everybody else is in a higher risk group. So the take-home message here is that if there is a child with sickle cell disease who has siblings, they should be HLA typed. And early on, the conversation should be had with the family about the option of bone marrow transplant from a sibling donor. Any other type of transplant today would still be considered experimental and should be done in the context of clinical trials. And so that is a consensus recommendation that young patients with an HLA matched sibling donor should be offered transplantation. I don't totally agree with the language of the European BMT group. I wouldn't say donor should be transplanted, but I would say should be offered transplantation as early as possible, preferably before school age, and then bone marrow or umbilical cord blood is the stem cell source of choice. Now, we talked about transplantation in young children, but increasingly, transplantation is being done in adults with sickle cell disease with good outcomes. And this is one paper from the STRIDE-1 pilot study, which showed that overall survival, and this is a combination of matched siblings and unrelated donors, had a 91% overall survival and an 85% event-free survival. Clinical trials have been ongoing from unrelated donor transplants as well, because only about 25% of the patients at most are going to have matched sibling donors. The outcomes are not quite as sanguine. When we used umbilical cord blood as a stem cell source, the outcome was not acceptable because of graft rejection, and that was stopped. Of patients receiving bone marrow transplant on the BMT-CTN 0601 study, overall survival was 86%. It wasn't in the 90s. And the cure, that is event-free survival, is 76%. So which means that 25% of the patients are not going to benefit one way or the other from this. Again, the risk of extensive chronic graft versus host disease of 38% makes it a little bit problematic in terms of the consequences and the duration of treatment. And patients had neurological complications that were reversible, but occurred in a third of the patients. So the current status of unrelated donor transplant is we are trying to improve outcomes. There are pilot studies that have been done to decrease the rate of graft versus host disease and improve engraftment. The next type of transplantation was haploidentical bone marrow transplant. Every person has a potential haploidentical, which means a half-matched donor. And there are several novel approaches that have been implemented in clinical trial for half-matched donor. Here are some collection of the case series presented and published by a number of groups. And it is beginning to look encouraging that there's a 90% chance of overall survival. And that's pretty, 90% chance of event-free survival and a 96% chance of overall survival. So if these data pan out in clinical trials, then half-matched donor transplantation has a potential for application worldwide and in many different centers without the need for very extensive genetic manipulation of the bone marrow. So having looked at what are the outcomes of transplantation for sickle cell disease, I would like to talk about ethical principles in this context. We often hear the words used somewhat loosely, oh, this is ethical or not ethical to do. I'd like to start off by saying that the ethicist can give you a framework to discuss an issue. They cannot tell you what to do. And what is the framework of biomedical ethics? So we look at non-maleficence, at first do no harm. We look at beneficence and obligation to do what is more likely to do more good than harm. Autonomy, that individuals have the right to make their own choices and make their life plan. And justice, that is distributing health resources fairly. In this context, avoiding anything which is unnecessarily or unjustifiably harmful. Almost anything that we do in medicine has the possibility of causing some harm. So the context is that, is it unnecessary or unjustifiable? Again, I'm not trying to give the answers as no ethicist can, but this is a framework to think about non-maleficence. On the other hand, withholding a potentially life, organ, and quality of life saving treatment is not beneficence. So beneficence would say that we should consider transplantation for the appropriate candidate. And then autonomy, that it is not for us to say this person may or may not have a transplantation, because in our mind it is ethical or not ethical, but ultimately consider the patient's right or the caregiver, when the caregiver is a surrogate, to make his or her own informed choices. A big problem is justice. We know that racial and ethnic minorities and people of low socioeconomic status do not have the same access to transplantation, just like they don't have access to so many other treatments. They also have worse outcomes. So access to healthcare unrelated to socioeconomic status, social capital, or political influence as an important consideration. And in this context, we must think not only in the wealthy parts of the world where we live, but also about the rest of the world. So having talked a little bit about ethics, I'd like to say, what do we know about cost effectiveness? Well, there has not yet been very good studies looking at cost effectiveness in the context of sickle cell disease for transplantation. But here's an idea of what these new drugs that have just come out in the last few years, crizanlizumab, voxelotor, and L-glutamine. And as you can see, the life expectancy with these drugs is, when you compare to the non-sickle cell population, and the population, US population in general, is fairly small. It is real, but it's fairly small. What is the estimated cost of disease-modifying therapies? And this is from the ICER data from last year. And as you can see, the annual wholesale acquisition cost, it's annual means recurrent. Again, as you can see here, we're looking at for crizanlizumab at $132,000, $127,000, and $40,000 for L-glutamine. Again, hydroxyurea wasn't in this mix, it's much smaller. So we would have to discount at these high, steep percentages to reach a threshold price of $150,000. But what do we think about the cost and cost effectiveness of transplant? And this is, again, not in the context of sickle cell disease, and the cost effectiveness and the types of cancer in terms of what is the total cost of transplantation. If you have a mean cost of $550,000 and a median of $408,000, with some conditioning regimens being less expensive, and this is autologous transplantation, again, not done in the context of BMT, that's more on gene therapy. But as you can see, this 408 and 374,000, when you compare to what your annual costs are, it suddenly starts becoming interesting to consider this and study this better to do cost effectiveness analyses. Let's talk a little bit about the rest of the world. And I picked India, well, just because I happen to know a little bit about India, and I happen to be sitting in India as I talk to you about this. So there's this notion that sickle cell disease is milder in India. Several years ago, Dr. Jain and others showed that it isn't necessarily so, that when you actually do good studies of disease severity in India, that your pain crisis rate are comparable, if not worse, than that reported in the cooperative study of sickle cell disease in the US, as is anemia, as is acute chest syndrome, as is stroke. So clearly, there are many people in the developing world who have just as bad sickle cell disease as we might see in the developed world. Well, what is there about cost effectiveness? And Dr. John from India published this, that the incremental cost per quality adjusted life year with the use of transplant versus chronic transfusion was $986 in the case of matched sibling donor transplant, and $2,579 in the case of unrelated donor transplantation. So the cost of transplantation for a matched sibling donor transplant in India may be anywhere between $10,000 and $20,000 compared to the $400,000, $500,000 we talked about. So they concluded that the probability of match-related donor transplant to be cost-effective, and I just want to point out that they had done these studies for thalassemia. Again, thalassemia, gets more attention in India than sickle cell disease is. But the willingness to pay is almost 90% at the threshold of Indian per capita gross domestic product. The interesting thing is availability of bone marrow transplant in the developing world brings additional distortions. So people, for instance, from Africa and from the Middle East are willing to travel to India to have a transplantation at the cost of a fraction of what it would cost in the United States. But there are very few Indian patients who are at the bottom of the social ladder who actually get a transplant. There are many possibilities of philanthropic support, GoFundMe support for bone marrow transplant for sickle cell disease in India. This is already happening in thalassemia. So I'd like to summarize that sickle cell disease is associated with significant risk of morbidity, impaired quality of life, and mortality. Transplantation from a matched sibling is associated with significant likelihood of benefit. Transplantation from unrelated, haploidentical, and then as my wonderful next speaker will speak on autologous gene therapy, these are all in clinical trials, and particular attention must be given to informed consent. And it is it behooves us to ensure access to those with limited resources to curative therapies. Thank you very much.
Thank you, Krish, for that presentation. Dr. Kanter, while she pulls up her slides, I want to remind the participants to put your questions in the chat box, and we'll answer those after the second presentation.
All right, can everybody hear me and see the slides? I'm going to assume yes.
Yes.
Thank you very much for having me today. I really appreciate it. I'm excited to talk about progress in gene therapy for sickle cell disease. These are my disclosures. So I'm hoping that I can better explain the different types of gene therapy and that the audience will have an increased understanding of how we measure efficacy in gene therapy, which is important because it's different between different gene therapies. I also want to make sure that the audience can understand the possible risks and causes of risk in gene therapy. And then we can start to understand what a cure for sickle cell disease could look like. So with that, we'll define the problem briefly that I think the audience is well aware of. We'll explain what gene therapy means, go through the types of gene therapy in use, what makes the different types of gene therapy different, and how we measure success in gene therapy. Then we'll discuss briefly how we define a cure for sickle cell disease and where we think the future lies. So, as most people in this audience know, sickle cell disease is the result of a single point mutation that causes a change in hemoglobin structure. And the result, of course, with sickle cell disease is pain, end organ damage, and shortened lifespan. And while we used to think that this was a simple disease of red blood cells that get sickled and stuck, we now know that's really not the case. And in fact, it's a far more complicated process that involves inflammation and adhesion, as well as ongoing hemolysis due to the fragile nature of the cells that contain sickle hemoglobin that can actually hemolyze long before they even sickle. The result of this disorder is multi-organ dysfunction. Unfortunately, there is not an organ that doesn't get affected by sickle cell disease in some capacity. And so later, as we discuss cure, it's important to think about all of these different organs and functions. So if sickle cell disease is caused by a mutation in a single gene, then we should just need to deliver a functional gene into the patient's cells. Seems like it should be simple. Gene therapy of autologous cells will completely circumvent the need for finding a matched donor and should have less or no immune-mediated side effects compared to allogeneic stem cell transplant. So what do we mean when we say gene therapy? Well, gene therapy is designed to introduce genetic material into cells to compensate for abnormal genes or to make a beneficial protein. It is an experimental technique that uses genes to treat or prevent disease, and of course may allow us in the future to treat a disorder as we are trying now by entering into the cells or altering a gene instead of drugs or surgery. So when we think about gene therapy, all of the different types of gene therapy that we're going to look at use the same overall process. They are made from autologous stem cells or the patient's own stem cells. We obtain these stem cells mostly through mobilization with apheresis, but can also be done by an actual bone marrow harvest. And those are the patient's own stem cells. Then we're going to discuss the different types of techniques we use to either introduce or increase production of healthy hemoglobin, whether that's gamma globin or beta globin, from those stem cells. And of course, we then give some form of chemotherapy to clear space and transplant the cells back to patients. Using this method, patients can serve as their own donor. It can be available for all patients, and there should be no need for immune suppression or any risk of graft versus host disease. So what types of gene therapy are currently being used? So the one most people have heard the most about is a gene addition therapy. This is where we add a new gene using a viral vector to deliver a non-sickling globin gene to the stem cells. So why a viral vector? Well, a virus is chosen as a vector because it already has all the programming it needs to get inside the cell. but we take out the viral genes and replace those genes with an anti-sickling gene. Using this process, however, does not remove or change any of the existing genes within the individual. This is a concept of what it looks like. Here's your viral vector, and here's the corrected gene. Now, it's important that different types of gene therapy are using different corrected genes. The two currently in use, one is a Bluebird Bio modified anti-sickling beta globin called HGB-T87Q that produces human hemoglobin A, but with a slight different mutation that both allows it to be better quantified and also gives it a little anti-sickling help. The other currently being studied is a modified gamma globin. This is a novel, highly potent variant of fetal hemoglobin, and it can be identified from endogenous fetal hemoglobin using this marker as well. So what about another type of gene therapy? Well, let's talk about gene editing. As it sounds, this edits a gene that's already in the stem cells. This can be done in a multitude of different ways. It can be done by removing, disrupting, or correcting faulty elements of DNA within a gene. It's done using an enzyme to cut the DNA at a very specific location. And we do that using a high precision lead or guide. There are a couple of different current trials using this technology. In some trials, we're reactivating protective fetal hemoglobin. So we're turning on an off switch. And in others, we're directly correcting variants that cause the disease. This can also be termed as gene correction. So this is one way of looking at this. And there's two main types of ways that we do this. One are using zinc finger nucleases, and the other is using CRISPR. I think most people have heard more about CRISPR. When you think of CRISPR, CRISPR-Cas9 has two components. It has a guide RNA that specifically matches the target site. That's your CRISPR part. And then it has a Cas9 nuclease that acts as a DNA cutting tool. If you look at the diagram over here, you can see that we use some different ways that we're going to talk about to get this guide RNA and Cas9 into the cell. Once it's there, that guide RNA identifies a very specific part of the DNA where it then cuts, and it makes a double-stranded break in most CRISPR-Cas9 trials that are ongoing. Once it's cut, new genetic material is filled in, disrupting the gene that was there. Another way of using CRISPR-Cas9 is a BC11A trial that is ongoing. Here, instead of using the same methods before, they use a lentiviral technology, and it inserts silencing equipment, if you will, or a silencing CRISPR-Cas9, or in this case, a small hairpin RNA, to disrupt the BC11A gene. The BC11A gene, by being disrupted, will turn fetal hemoglobin back on. Finally, we have another type that's a combination of gene editing and addition. In this scenario, you can edit a gene that's in the body and at the same time put in a new oligonucleotide strand to produce a new gene product. This is known as HDR, or homology-directed repair, and it requires a homologous DNA strand that is used as a template for high-fidelity DNA repair. What does that look like? So here is another diagram of CRISPR-Cas9. And remember in group A, in the field A up here, how I explained that you're going to insert a guide RNA with a Cas9 cutting enzyme. And when it makes a cut in the DNA, it makes a double-stranded break. So in one possible mechanism, this causes this break in DNA and what we call an indel formation. That's where we have the DNA repair itself, essentially repairing itself incorrectly. And that is what we are using to quantify efficacy in these types of trials. Another version, however, is homology-directed repair. And there are a couple of different ways this is done. The same DNA repair can be, the same DNA break, that double-stranded DNA break is made. But now there's an extra strip of DNA that's already made, and it comes in the same complex with the CRISPR-Cas9. And what it does is automatically fills that space because it is perfectly made to fill the space that the cut was made in. So in this case, you could cut out the actual mutation of the sickle mutation and fill in with a gene that makes healthy beta globin. Finally, there's some also newer genome editing approaches that are using components from the CRISPR-Cas9 that I just told you about, but using a different enzyme other than Cas9. This allows a specific point mutation as opposed to that double-stranded break that we just talked about. Here, we're using what we call DNA base editors. These comprise catalytically disabled nucleases fused to a nucleobase deaminase enzyme. In other words, the same type of guide RNA is used, binds to an identified location, but instead of a double-stranded DNA cut, this single enzyme makes a one-pair change and can change from this match to this match, obviously altering the DNA and the various proteins that it encodes for. This is called base pair editing, and I think we will see it soon in clinical trials. So getting new genes or the gene editing CRISPR-Cas9 tech into stem cells is not easy. One way of using this, as I mentioned, is a lentiviral or viral envelope. In this scenario, we take a virus and we take out, in this case, a lentivirus, we take out the instructions for making HIV. Instead, we have our empty envelope in which we put the new gene for hemoglobin. That, of course, gets sealed up inside the viral vector and made to deliver to the stem cells using pieces of other viruses in the meantime. Another way of doing this, however, is called electroporation. Most of the gene editing technology uses electroporation to get the CRISPR-Cas9 tech into the stem cell. This is a microbiology technique in which an electrical field is applied to cells that increases the permeability of the cell membrane, allowing chemicals, drugs, or in this case, DNA or DNA technology, to be introduced into the cell. In layman's terms, we shock the stem cells that allows the CRISPR-Cas9 and, if needed, oligonucleotide strands to get into the stem cell. So when we're thinking about efficacy in gene therapy, it's important to recognize how we speak that language. So the first is looking at vector copy numbers. Remember, we talked about that a vector is used to bring in that new gene. By looking at a vector copy number, it can tell us how many of those gene therapy letters were delivered to a sample of blood cells. This is important because we want to make sure, as much as possible, that all of our stem cells get a letter or get a new gene. So we prefer a vector copy number of 1 or above. Transduction efficacy is the percentage of blood stem cells that have been successfully transduced. So if you had all of the available stem cells, we'd like as close to 100% transduction efficacy. And then, of course, we have cell dose, the amount of a patient's own blood stem cell that's returned to the patient after gene therapy. This same language is used in gene editing, but instead of vector copy numbers per cell, we talk about edits per cell. And instead of transduction efficacy, we talk about editing efficacy. And the same language can be applied. So when we think about stem cells, it's important to remember when we talk about vector copy numbers, we're talking about an average. In other words, if there are 4 letters or 4 new genes, 1 in each of 4 stem cells, our VCN is 1, and we have 100% transduction efficacy. This is truly our best case scenario. In other terms, however, sometimes 2 genes per cell could be placed, but some of our stem cells don't get the gene at all. In this case, the vector copy number is still 1, but our transduction efficacy is 50%. So hopefully you can see why we'd rather have this scenario on the left than the middle. And then in the worst case scenario that I have here, there's only 1 gene delivered to 1 of the 4 stem cells. This leaves a VCN of 0.25 and a transduction efficacy of 25%. The exact same terminology can be used when discussing transduction efficacy, or in other cases, editing efficacy. Once all is said and done, we now need to make sure the new hemoglobin gets where it's supposed to, whether it's a beta globin or a gamma globin, so hemoglobin A or hemoglobin F. We want to make sure that it's evenly distributed in all of the cells. This is one example of an exploratory assay that can allow for single cell resolution of hemoglobin expression. In other words, this can help determine, in the case of Bluebird Bio, is used to see if their new hemoglobin, A T87Q, gets into every red cell. When you think of an individual who does not have sickle cell disease, they have hemoglobin A throughout all of their red cells. Somebody with sickle cell disease has hemoglobin S throughout all of their red cells. In someone with sickle cell trait, they have A and S in every single one of their cells. It is why, for the most part, they are protected from any complications because they have that hemoglobin A. However, if you think about somebody we transfuse, now they have S cells and A cells that are separate. And when we transfuse someone, their hemoglobin from the new cells doesn't get into the old ones. Our goal with lentiglobin and other gene therapies is to create something that looks more like sickle cell trait, where you have a pancellular expression of, in this case, T87Q. But the same would be true if we were talking about other hemoglobins or gamma globins. So what are the risks? They're slightly different between gene therapy and gene editing, but also have very high concordance. Gene addition therapy using a viral vector allows a random vector insertion. Now, the vectors are programmed to insert in certain places, but it's not 100%. The good news is our current technology allows us to monitor insertion sites to assess any that are developing what we consider clonal dominance or superiority that could signal a problem. And the risk of an insertion at a promoter that could, of course, cause an unwanted cellular proliferation. In addition, these individuals are all receiving chemotherapy, mostly myeloablative, and there are associated risks. There is the risk of using a viral vector. While we think this risk is probably the lowest on this list, it is still a possible risk. And there are stem cell abnormalities that could be present in the stem cells themselves before they undergo transduction or editing, and that could be enhanced by the process of transduction or electroporation or exposure to a chemotherapy ablated bone marrow. And then we have to ask if the bone marrow niche is conducive to optimizing outcomes. Thinking about gene editing, you can see very similar risks. Gene editing can also result in unintended modifications at other points along the genome. These are called off-target effects. Now, current technology allows us to identify off-target mutations that occur at high frequency, but it's possible that more rare ones could escape detection and give cells growth or survival advantages that could also promote a malignancy. These technologies also require chemotherapy. And as mentioned, with electroporation, there's a decreased stem cell survival and more donor stem cells or patient stem cells are needed. And as we just discussed, there are stem cell abnormalities and problems that are possible within the bone marrow niche. So thinking about the recent patient that's made the news from Bluebird Bio's 206 or long-term follow-up trial. This individual was 5.5 years out of gene therapy and developed AML, or acute myelogenous leukemia. As we're evaluating the cause of this individual's AML, we have to think about, was it the chemotherapy that she received? Was it the busulfan? Was it that we gave her busulfan, but we didn't ablate all of the stem cells, and instead, those stem cells that were actually exposed to busulfan but recovered are now injured? Or could it have been a vector problem? Could the vector have inserted itself near a promoter? So how do we ask these questions and elucidate these answers? We need to ask what type of AML it is. We need to ask if it developed from stem cells that have a CD34. We need to see if the stem cells have a vector in them, as they actually do in this case, or in gene editings in other case. In other words, are these transplanted stem cells, or are they native stem cells that recovered from the chemotherapy? We need to know if it looks like a typical AML. Is there a new cytogenetic variant that could tell us if there was an insertion site problem? And then if the malignant cells are from the donor or transduced or edited cells, we need to know, is the problem the editing or the transduction? Was there a process that's undergoing the manufacturing that's causing a problem in the stem cells? Or is there an issue with the bone marrow niche? This individual had stem cells obtained directly from the bone marrow, underwent bone marrow harvest, and not plexiform mobilization. So we have to ask specific questions as whether we think this could be a bone marrow specific problem, or if the same problems can come from plexiform mobilized stem cells. And then finally, when we think about a cure or a grade of cure, we have to think about both the biologic definition. In stem cell transplant, as you heard previously, we have to look at the percent donor. If someone now has 100% donor stem cells, we know they can only make healthy hemoglobin. Well, what about in the setting of gene therapy? We have to make sure the new hemoglobin, however it's produced, whether it's fetal or beta globin, there's enough of it that prevents sickling. We also want to make sure that hemolysis is completely resolved. And we want to look at the rheology. How does the cell act, move? Does it adhere to the inside of the blood vessel? It has to be at least equal to that of sickle cell trait. And that includes looking at endothelial adherence and inflammatory stimuli. But we don't just care about what it looks like under a microscope or in a laboratory. We also have functional definitions of cure. Does it completely rid individuals who are affected from pain episodes or fatigue or other important patient-reported outcomes? Further, we have organ-based definitions. Remember, sickle cell disease affects multiple organs and could cause multiple types of organ dysfunction. So what we need to understand from gene therapy is, does it just stabilize some of the organ dysfunction that was already there? Does it prevent it from worsening? And if it does worsen, what amount of worsening organ function is still acceptable to be considered a cure? We have to define this grade of cure in the long run. We also have to ensure that we are honest with patients about the options, the risks, and the outcomes. We have to monitor long-term, not only to look for side effects or complications, but also in defining cure. We have identified that potential issues could be inherent to stem cells or the bone marrow niche in sickle cell disease. And if that's true, how do we identify the healthiest stem cells? And how do we improve the bone marrow niche? And then we have to figure out, as well said by Krish, how to make this type of therapy available, affordable, and universal. So we covered why we were using gene therapy, what types of gene therapy we are using, how we know if gene therapy is working, and the different ways in which we need to measure gene therapy, the risks of gene therapy in sickle cell disease, and hopefully at defining cure in the future. And I think Krish and I are now happy to take any questions that you have. Thank you again for having me.
Thank you to both speakers. Really, really wonderful presentations and a nice information for us to have some discussion. Jessica, do we have any questions in the chat box yet? You're on mute.
The question that came in actually from you, Dr. Pace, what's the youngest age for Dr. Krish? What's the youngest age to transplant if a matched sibling donor is available?
So I'm happy to expand on it briefly, if you would permit me.
Of course.
So why is the youngest age important? So youngest age is important in some conditions, right? So if you have a rapidly progressive metabolic disorder, then you want to, intervene as early as possible. So if you had like a Hurler syndrome, getting the transplant done before age 2 is really important. Is a sickle cell disease a rapidly progressive disorder? Well, the risk for stroke starts picking up at around age 2. And certainly, adding hypoxemia on a continuous basis and progressive vasculopathy is not a good thing. We do know that under the age of 5 years, the outcomes are best. So I think that we started transplant, all those years ago as a treatment for the worst of the worst, right? So we were only going to transplant people who had a stroke, they're high risk of dying. And then we also started transplanting adults. And so early on, we were transplanting people with such bad disease that they weren't doing well. And so as the treatments got better and we had better proof, we kept doing transplants at an earlier and earlier age. So the other thing is, if there is an older sibling, I mean, for the crazy period of our lives when we think, having children is a good idea, our children are fairly closely related in age. So it's quite possible that a 2 or 3 year old, is big enough sort of, in terms of the airways and blood vessels and, simple things like that, may be considered for a transplant if there is a sibling. So it's not uncommon in our program to have 3-year-old children undergo transplant from a matched sibling donor. We have, and those children do really, really well.
And what's the youngest age for the sibling donor? Is there an age or a size?
So the umbilical cord blood is the youngest age, right?
Right, right.
So the umbilical cord blood, again, is a set volume. So generally, the children will outgrow it by the age of 3 years. Now, we now have the ability to expand the umbilical cord blood. So the future, has all kinds of possibilities.
Mhm. But what about not umbilical cord blood, a sibling? What would be the youngest if
Sibling
you had a
donor,
matched sibling?
again, you know, across the board,
Okay.
you know, people 8, 9, 10 months, 1
Okay.
year, you know, like if
Okay.
you're about 10 kilos,
Okay.
like my mentor used to say, when you can measure children like this, not like this, you know, I
Gotcha.
think that they're easier to use as a, yeah.
Wonderful.
We have another question from Dr. Steve Goodman. He thanks you all for the wonderful talks. What are the approaches for targeting gene editing machinery?
I'm not sure exactly. I could use a little more clarification, I think, on the question in terms of targeting gene editing machinery. I'm not sure what the individual means by that. I'm sorry. We have lots of options there. We could talk about how they use gene editing targets, or we could talk about improving the CRISPR-Cas9 methodology. And I think that the next sort of era of this is going to be looking at base pair editing.
Nanoparticles.
Using nanoparticles. I wish I knew more. Yeah, I wish I knew more about nanoparticles. I think the next place we're going is base pair editing. And I think there's some really incredible options when we look at the base pair editing. It is 100% on target, at least so far in mice. And it may give us the option for direct editing of the sickle mutation. So I'll have to follow up with you on the nanoparticles. I apologize.
May I take a stab at it, Julie? So nanoparticles are a means of delivering the genetic material, right? So when you get your Pfizer or Moderna vaccine, you're getting a liposome. So there is some experimental evidence of using nanoparticles for delivering some gene editing. In fact, the group at Yale has done something in thalassaemic mice. So that there is a possibility for using nanoparticles to deliver. And then all of these also may be relevant in vivo gene therapy, which I'm sure Dr. Kanter can talk a lot about it, that could happen in the future. Oh, Dr. Lee said the same thing.
So Julie, I'm not sure I even need to relay these to you because you can read them, but we do have another, so that the rest of the group can hear. Can you speculate on what would be direct in vivo gene therapy?
I think that's a great question. Thank you, Koff. That's really what we're going to need to look for to make this a universal option. I think right now we definitely aren't there yet. We need the technology that would allow us to in vivo edit cells that wouldn't be immediately rejected by the body. And I don't think we've figured out how to do that quite yet, or at least I certainly don't know of anyone who's figured it out. I know that the closer we get to something like a base pair edit, the closer we will get to the option for in vivo editing. But I think the real question is going to be, how do we not just edit, but make sure that the stem cells don't get cleared? Someone recently asked me if we can use an mRNA vaccine, right, to treat COVID. Why aren't we doing that in sickle cell disease? And I thought that was an excellent question. And the answer is because mRNA does code for protein, but it's very short-lived. So it would code for, in this example, we could make an mRNA that would code for beta globin, but it wouldn't live very long. And mRNA does not stay in the cell. It's what makes it such a great vaccine. It doesn't cause any permanent gene editing. And instead, we have to figure out a way to do something similar, but with a gene edit that can stay in the cell.
Yeah, and if I may just take a stab at it, I'm sorry. So Peter Drazer's group at Yale is actually has used these nanoparticles in vivo, actually in utero. And so if you can somehow get trophism to the hematopoietic stem cells, and then you can deliver the, a genetic editing machinery there. And just recently, Novartis and, the
Gates Foundation.
Gates Foundation. Yeah, I think they both have a little bit of, loose change lying around. And this is a powerful partnership targeting in vivo gene therapy. So I hope that in the future, this will put the bone marrow transplanters out of business, and we'd be able to do this directly in vivo.
Actually, that's a great segue to the question that I have. Now, bringing us back to reality and what we do for patients right now. A lot of those techniques are wonderful and exciting, in the laboratory, but for patients, and Krish, maybe you could start and you can chime in as well, Julie, how do we talk to the patients about all these choices and not get them confused? What is really available now, traditional bone marrow transplant versus going for gene therapy, which also involves, some form of transplantation as well. How do we talk to patients about this, and especially in light of the recent halting of the Bluebird Bio trial?
I would say this in 3 words: early, often, and with absolute honesty. And this is how we do it. People, even in the last 15 years, there's a change. People would come for a transplant consult, dragging and kicking. They came when all of the doors were closed. Now people come and say, I want to find out all my options. I want to find out about quality of life. And I think that regular people can digest complex information when given to it in context without trying to demonstrate our erudition and learning.
I think that is absolutely correct. I approach this in a sort of a streamlined way, starting with looking at the individual, looking at what they are looking for, what complications do they have now, and what are they hoping to achieve. If they have a matched sibling, I always and every time, no matter their age, recommend a matched sibling donor transplant, because it's what we know the most about and have the most information on. That being said, I think once we get into looking at time away from home, time away from work, there is a very big difference between allogeneic and gene therapy. And having done both, especially in adults, I think that that's something we're going to have to think about. My patients in gene therapy recover much faster. There is no risk of graft versus host disease. They need much less frequent follow-ups. People have said in the recent discussion about the patient with AML that I am biased. I would admit it. I absolutely am biased to wanting gene therapy desperately to work because it is so well tolerated. And while haploidentical transplants for those without matched sibling donors are possible, they are much more difficult in the short term. The other thing that I think is important to recognize is that the risk of leukemia is not just in gene therapy. And in fact, we have seen several patients who have failed haploidentical transplants that also had leukemia, which is why I think it's so important that we better assess the stem cells themselves, the risk of ablative versus non-ablative chemotherapy. What's the risk of exposing stem cells to chemo that get left behind? Is there more risk in that scenario? And I think these are really important questions we need to ask.
If I may follow up this wonderful response from Julie. So we have haploidentical, mismatch unrelated donor, matched sibling donor, comparative trial, 6 gene therapy studies. And people ask me, so how do you prioritize? I tell them it's not for me to prioritize. I am not the one making the choice. And so it actually takes a little while of doing this to understand how to speak and how to walk people through. As an example of how people are able to understand complex information, I was speaking to a family with a Spanish interpreter who had never had a consult for bone marrow transplant. They thought that they had to bring a donor from the street somehow who's ready to give up their bone marrow on a one-way donation. 15 minutes into the conversation, the mother said, oh, what's that you said where we can get your own cells back? I want that. And I say it as an example that absolutely with confidence, if you take the time and keep it straight, people can understand. I do understand that doing both, that, you know, not having allogenicity, my social worker says, oh, it's a gene therapy, it'll be fine. I mean, anybody in the staff can see that, but the risks are real. And, you know, 20 years ago, we got in a whole bunch of trouble, you know, with cancer. So folks like us who are investigators in gene therapy studies are saying to the sponsors, we're going to heat, you know, we're going to hold your feet to the fire. And you have to tell us not just that the gene therapy didn't cause this, but what caused this. And the final thing I want to say, which is really difficult in a transplant center, is who do you tell that you have an option? Who do you say is the right candidate? I just say anybody who's suffering from sickle cell disease. I have a problem with saying, well, this person, you better not tell them the information, because I don't see that individual clinical situations, individual priorities are immutable. And you have an honest conversation. And many times people who are stuck in that cycle are never wanting to get out of it. But I would have this conversation with anybody who's willing to have the conversation.
Looks like we have one more comment in the chat box, Jessica. You're muted, Jessica.
Just the comment. Oh, yep, one more. What about these options in the contest of Africa? When will we have these complicated therapies there?
And I think that is such a great question. I know there are centers where you can undergo hematopoietic stem cell transplant in Africa, in Ghana, but I think it needs to be an option that is far more widespread. And I don't think we have a timeline. And I don't know that we need to wait for in vivo to make this a reality in other places. We just need to make it safer, and we need to better understand what's the minimal care sort of needed around taking someone through this process. And so I think we're working on how we do that in a most optimal way. And I think one of the things that makes gene therapy viable in lower resource areas is the less risk of graft versus host disease, which needs more complicated monitoring. But we're not quite there yet until we can make sure it is truly safe and that we can optimize all of our outcomes, I think.
Thank you. If I may add, you know, there are centers, for instance, there is at least one center in Nigeria that's reported a mad sibling donor transplant. So there are people who have large families. I mean, and there is subsidized HLA typing done. So some of it is the infrastructure has to get better. You know, I mean, that is what it boils down to, having infrastructure for testing, infrastructure for, you know, the care and all of that. Today, people in Africa, there are multiple physicians who travel to India and are licensed in Africa and function in transplant centers in Africa. And the patients go to India, just as an example. They could be going to Thailand, they could be going somewhere, a fraction of the cost. But ultimately, going somewhere else is not a solution. There are people in Africa, you know, and then a lot of us are very committed to training physicians from Africa. So ultimately, we have to not train only the physicians, but the entire infrastructure. And remember that the Gates Foundation and NHLBI partnership is ultimately about bringing gene therapy to Africa.
So that is a wonderful, wonderful note to end on. And the last comment I will make from the chat box is from Koff. He says he can't wait to make this a reality. So perfect. Thank you, Koff, for that comment. So we're right at 2:30, and I believe this will be the end of a webinar. This has been wonderful. I want to thank both of our speakers, Dr. Kanter, Dr. Krishnamurti, for a wonderful update on bone marrow transplant in general and gene therapy. If there are other questions, I guess people can send them directly to Jessica, but thank you all for participating today. And thank you to the audience as well.
Thank you all so much.
Thank you. A pleasure.
And as a note, we will post the recording of this event live on the SCBM website. So if any colleagues missed it, or if you would like to view it again, please visit scbm.org, and you can also find contact information for me there should you have any follow-up questions for today's speakers. Thank you all again so much, and thank you for attending.
Thank you.
Bye.
Bye.
Take care.